Okay, today I'm here to talk to you about my research this summer. This summer I was looking at various metrics that will help us better understand our oceans, and we see CMIP6 data in the first plot, which covers multiple ocean models, and I looked at three boxes in the ocean, one in the Pacific Ocean and two in the Atlantic Ocean. Um, the first plot, we see future minus present data for a bunch of different variables and a bunch of different models. We looked at biomass, chlorophyll, diatoms, and zooplankton, and by plotting these differences, we're able to analyze the seasonal cycle, and we can notice how they change throughout the year. For example, we see how phytoplankton populations peak in summer months after the phytoplankton bloom, which is an expected result. We also then further analyzed one of the variables, which is SpCO2, which is the surface aqueous partial pressure of carbon dioxide, and that is the plot on the right. What we're looking at is the separations in the temperature dependent component and the non-temperature dependent components of this variable. And by doing this, we can see how it's affected by biology and temperature, and separating it creates these plots that allow us to analyze these differences. One other thing that I did here was I managed to add observational data so that we can compare it to the model's data. And we see that it's the black line, which is with filled in black circles. And what we noticed is that the observational data falls within the deviations of the model data. And this is good for us because not only can we now look at these plots to analyze the fluctuations throughout the year, but we can also now know that these models are reliable, especially with SpCO2, because they are very similar to what was recorded in the fields. So that's pretty much what I did this summer. Thank you for listening, and thank you, Curve.